Today we'll be talking about the process of transcription. The DNA performs two major important functions. One is the transfer of genetic information from the parent to the offspring, and this is achieved through replication, where DNA copies onto itself similar, and it is known as replication. It is a autocatalytic activity because here the DNA is producing or is just developing or making an exact copy of itself. The conversion, or where the process where the DNA gets converted into RNA, it is known as transcription, and it is an heterocatalytic activity. So we can define the process of transcription as the transfer of or genetic information from one strand of DNA into RNA is known as transcription. Now here also the rule of complementary is there. We can explain it. Like in the case of DNA to DNA copying, we have adenine pairing with thymine and guanine pairing with cytosine. In this case, that is DNA to DNA. But in the case of DNA to RNA, adenine will pair with uracil. That means the thymine is replaced by another nitrogenous base which is known as uracil, which is found in RNA, and guanine will be pairing up with cytosine. So the complementary base pair also governs over here. Only thing is that the thymine is replaced by another nitrogenous base which is uracil. Now a question can be asked, like, how is replication and transcription different majorly? Now in the case of replication, both the strands of the DNA of an organism are duplicated. So over here in the first case, both the DNA strands will be duplicated and the genetic information will passed on as such to the offspring, and the parental DNA act as a template. That is, we were talking about the replication process. When we talk of the transcription, in transcription only a segment of the DNA and only one of the strands of that DNA strand is copied. So there is a difference between the replication and transcription, because in the transcription process our main motto is to get the RNA, not the DNA, not the transfer of complete genetic information, because RNA eventually leads to the formation of the proteins. So over here in the case of replication, we have the complete DNA being transferred, that is, the copy is being made. But over here in this case, only one of the segment, and out of that only a portion of that segment is being copied. So the replication and transcription are quite a different process. Now a question can be asked, why this is so? Why only one strand is copied and not both the strands are copied during the process of transcription? Now the first reason we can give is, if both the strands act as a template, then they will each code for RNA and with a different sequence. Because remember, the DNA sequence are complementary to each other, they are not exact. So what is there? As a result, the sequence in the protein will be different and they will be coding for different kinds of amino acids. You can understand it because if we talk about the two strands of DNA, they are complementary to each other, but at the same time they have different base pairing. So it is AT over here, let's say GC over here. So what will happen? This will code for a different protein and this will code for a different protein, because protein that is code, it is a code of triplet. So what happens is, it will create a lot of confusion. So therefore only one strand is being coded, and out of that one strand only a portion of it is coded for a particular type of a protein. Second, if both the strands of DNA are used for transcription, it will produce two complementary RNA strands. Now what is happening over here? Another problem is, if we know that the DNA strands are complementary to each other, now if both are copied, these two strands will also be complementary to each other and they will form double-stranded RNA. So the entire purpose of transcription will not be achieved. So what will happen if the double-stranded RNA is there? It will not be translated into protein and the transcription process will not happen. Therefore, it is very, very important to have that copying of one DNA strand or a portion of one DNA strand which is being then transcribed, or transcription takes place through that unit. Now let us talk about the entire, when we are talking of the DNA strand being transcribed, undergoing transcription, there is a certain area which is known as the transcription unit. And it is that transcription unit which actually takes part in the process of transcription. So if we see, the transcription unit in the DNA has three major parts. That is, first is the promoter, the second is the structural gene, and the third is the terminator. That is, you can remember it by PST, that is promoter, structural gene, and the terminator. Now when we talk of the promoter, a promoter actually is a specific sequence of nucleotides of DNA. So it is this promoter, it is a specialized sequence of DNA, and it is here the RNA polymerase binds for transcription. So at this promoter, it's a special sequence. It's a special sequence, and RNA polymerase enzyme binds here only due to its specialized sequence. So the RNA polymerase enzyme will bind over here, and it is located towards the 5' So you can see over here, it is located towards the 5' of the coding strand. Now when we are seeing the transcription here unit, you can see there are two units which we'll discuss about it later. The two strands, the one strand is known as the template strand, the other one is known as the coding strand. So this promoter will be present towards the 5' end of the coding strand. So 5' end of coding strand, the promoter is present. Next is the structural gene. When we talk of the structural gene, now you all know the two strands of the structural gene of the transcription unit are anti-parallel. That means they are having opposite polarity. When we are saying structural gene, this is entire structural gene, and this is having 3' to 5', 5' to 3'. The two strands will have opposite polarity. Now the main enzyme which is taking part in the process of transcription is DNA dependent RNA polymerase. This is the main enzyme which is taking part in the process of transcription. So what happens is, this enzyme can catalyze the polymerization, that means the addition of the nucleotides in 5' to 3' direction. So this works as we have seen similarly. The DNA polymerase enzyme also worked from 5' to 3'. The DNA dependent RNA polymerase also functions from 5' to 3'. So this enzyme attaches to the structural gene. Now the strand of the DNA, therefore 3' to 5' will act as a template. Now why we are saying this as a template strand? Why we are not saying 5' to 3' as a template strand? The reason is the DNA dependent RNA polymerase can have the polymerization of nucleotides in 5' to 3' direction only. Therefore, we cannot, this strand which is 3' to 5', it is known as a template strand. Another term for it is sense strand. That is, because it is through this we are going to have the coding for mRNA. Now, the other strand of DNA with 5' to 3' polarity, that is we are talking of now this strand. So, this strand does not code for RNA. Although it is known as the coding strand, but it does not code for RNA. Actual coding of RNA is this 3' to 5' and therefore we are calling it as a template strand. So, this coding strand is, since it is not coding for any of the RNA, it is known as antisense strand. So, template strand is equal to sense strand. Whereas when we talk of the coding strand is nonsense, or we can say antisense. Antisense is better, so we call it as antisense strand because this actually does not code for any type of RNA. And all the reference points we are talking of are made in accordance with the coding strand. So surprisingly, the coding strand, although it does not code for any of the RNA, but all the reference points we are, because the promoter, when we are saying it is toward, we are not saying it is present at 3', but it is present at the 5'. Again, the terminator is present at the another end, which is 3' end. So all the reference points we will be using will be according to the coding strand. Now, so over here we see 3' to 5' will be the template or the sense strand, and the other strand will be the coding strand, that is having a polarity of 5' to 3'. So if we can work it out accordingly, if the sequence over here is given, that is the template strand, the coding strand, we can get it. Now terminator, what is terminator? It is a particular sequence of nucleotides again on the DNA. Just as we had the promoter, it is also a special sequence of DNA that regulates the end of the termination or transcription process, and it is located towards the 3' end of the coding strand. It is also said to be the downstream of the coding strand because it is towards the end. So we have the transcription unit, which is mainly a double stranded DNA with a polarity of 3' to 5' and 5' to 3'. One promoter gene, which is a special sequence of nucleotides located at the 5' end of the coding strand, and also it is here the RNA polymerase enzyme binds. Then we have the structural gene, which is actually the site where the transcription process will be taking place, and this structural gene has we have defined it as a template strand and the coding strand. The template strand actually acts as a template where the synthesis, which codes for the RNA and is therefore known as the sense strand. The coding strand, on the other hand, does not code for any type of mRNA and it is known as antisense strand. But we all, when we are talking of various units or parts of the transcription unit, we always refer in context with coding strand. Now, the other important thing is terminator. Terminator is again a specialized type of DNA sequence that defines the end of the transcription process, and it is always located at the 3' end of the transcription unit, or we say the 3' end of the coding strand. Now besides this, there are other regulatory sequence. Now next part we are going to just try and understand that how does it acts. Now the promoter over here is very important and the terminator. So let us study about the actual process of transcription.